then she got a PhD in chemical biology from Harvard University, and she worked with a very well-known group, David Liu, studying RNA modifications, which is now a very interesting topic in the RNA field. And she's going to talk to us a bit more about RNA modifications, especially the work that she was doing with Howard Shank in Stanford. And RNA modifications in CircRNAs is in the connection that is being described now with immunology. And as a fun fact, Grace was once a guest on a kids program. I think it's in US television, which she was doing some chemistry expertise appearance. So that sounds pretty fun, and I'm very excited to hear your talk, Grace, especially because I'm particularly excited now with modifications. So thank you very much for being ready now for us, and let's start with your talk.
Thank you so much, Mariella, for the introduction, and to all the organizers for the invitation to speak. I will share my talk. Should be full screen, is that right? Okay. Right, so we have heard a lot about different circular RNA aspects today. It's a fantastic symposium. And I want to share about the ways that we are thinking about circular RNAs. So we may know that circular RNAs are expressed throughout the eukaryotic kingdom of life. This is a phylogeny tree, and I just labeled some of the species where circular RNAs have been identified, and you can see the diversity of species that are present. The metazoans are labeled in gray, sort of, right here, if you can see the pointer. Actually, one of the first indications that circular RNAs existed in nature was actually from a virus, the hepatitis delta virus, which has a circular RNA genome. And this electron microscopy image showed really beautiful, the circular nature of these structures. More recently, just in the last year to 2 years, people have found that oncogenic double-stranded DNA viruses, like the Epstein-Barr virus, the Kaposi sarcoma-associated herpes virus, and the human papillomavirus, also encode circular RNAs. So when I started my postdoc, we didn't yet know about these double-stranded RNAs, but we knew about the hepatitis delta virus, and we had the hypothesis that likely other viruses would encode circular RNAs. So given that a healthy cell needs to maintain this vital balance between recognizing pathogenic viral RNAs to prevent infection, while also recognizing its own self-endogenous RNAs to prevent autoimmunity, we had several different questions. How do mammalian cells distinguish between self and non-self circular RNAs? What are the molecular mechanisms to sense the circular RNAs? And can these distinctions be harnessed for therapies? So to answer the first question, we designed the system that used a group 1 intron. So this intron in its natural state typically splices itself into a circle to join the two ends of the exons together. However, if we take this group 1 intron, split it in half, we put the 3' end upstream of a sequence of interest and the 5' end downstream of a sequence of interest. We find that in this permuted intron exon system, whatever is in between these two intron halves will actually splice itself into a circle in an autocatalytic fashion. So this means that during in vitro transcription, without the addition of any other protein components, we can generate circular RNA. We call this a CircFOREIGN. During this in vitro transcription reaction and autocatalytic splicing, we also generate some linear RNA byproducts, which become substrates for an exonuclease RNase R that digest from the 3' end of linear RNAs in a very processive fashion to degrade them very quickly and efficiently. So the first thing that we did was we took this CircFOREIGN and we transfected it into cells, and we chose HeLa cells. And then we compared a linear RNA control. So this is a piece of RNA that has a exact same sequence as the circular RNA, but it has a 5' end and a 3' end. And on the 5' end, there's a 5' hydroxyl. And then briefly, I didn't mention in the last slide where the internal ribosome entry site that's present, let's see if I can go back here, that's present on the strand of RNA actually becomes upstream of the open reading frame, so that when we transfect in the RNA, we can allow and observe translation of this GFP. All right. So when we transfect in either the circular RNA or the linear RNA into cells, and then we collect the RNA and we perform RNA-seq, we get a plot like this, where each dot represents a gene that was enriched in either cells that received the circular RNA transfection or cells that received the linear RNA transfection compared to mock transfection. The dots are color-coded such that the dots that are more red are more enriched in the genes that are more enriched in cells that receive the circular RNA transfection compared to the cells that receive the linear RNA transfection. And so if we perform gene ontology analysis, we can see that the genes that are expressed in cells that receive the circular RNA transfection were highly related to the inflammatory response, defense response, response to cytokine. So we wanted to dig deeper into what was happening. We selected the genes I labeled in bold for QRT-PCR analysis. Because we used HeLa cells for the RNA-seq experiment, we started there. And so here, we transfected in either linear RNA control or the circular RNA. And then we measure this panel of innate immune genes that represent, so like a response to viral infection. And so we noticed that the cells that received the circular RNA transfection had much higher expression of these immune genes. So then we wanted to know if this was a generalizable phenomenon to other cell types. And so next we look at keratinocytes, which are skin cells represented by the Hackett cell line. So often the skin is the first barrier and the first to infection, and potentially infections can happen on the skin. And we found that the circular RNAs, when they were transfected into the Hackett cells, also induced a much higher immune response compared to the linear RNA transfection. We also looked at macrophages as represented by the raw cells, because those are the immune cells that usually respond to the site of infection first. And again, we saw a similar pattern where the cells that received the CircFOREIGN transfection had much higher immune response compared to the cells that received the linear RNA transfection. We wanted to know what was happening here, because as I mentioned at the very beginning, eukaryotes are expressing thousands of different circular RNAs, and those cells are not all undergoing inflammation or an inflammatory response. So we hypothesized that maybe proteins that are associated with self-circular RNAs may be different and distinct from the proteins that are associated with foreign circular RNAs. And so to test that hypothesis, we used a method called CHIRP-MS, which is the comprehensive identification of RNA binding proteins by mass spectrometry. So very briefly, we take live cells and then we cross-link them. So then we can covalently join an RNA with all of their RNA binding proteins. We then use biotinylated Thailand oligos to enrich for the entire complex. And then after some washing and elution, we can identify by mass spectrometry the collection of RNA binding proteins that are present on a specific target RNA. And so we took several different RNAs through this process. We took the CircFOREIGN as well as its linear RNA control. And then we also compared them to a CircSELF. So here in the CircFOREIGN, we use this foreign autocatalytic sort of self-splicing mechanism to generate the circular RNAs. But we know that endogenous circular RNAs are formed during transcription and splicing by protein-assisted splicing, so the splices of a complex. And so we took an endogenous intron set from the gene ZK-SCAN1, which typically produces exon 2 and exon 3 of ZK-SCAN1 in a circle. We swapped out the ZK-SCAN1 exons for a GFP IRES sequence. So here we're comparing the process that the cells are using to make the circular RNA while keeping the actual sequence of the circular RNA constant. So we found that when we analyzed all the proteins that are associated with either CircFOREIGN or CircSELF, there was a collection of proteins and enzymes that associated with the self, CircSELF CircRNA, but not the CircFOREIGN. And those are related to the RNA modification M6A or N6-Methyladenosine. So briefly, the M6A modification is a methyl group attached to the N6 position of adenosine. The enzymes that install the modification, we call them writers. There are proteins that recognize the modification, we call them readers. And then there are enzymes that remove the modification, and we call them erasers. And so we wondered, could M6A be a molecular mark to tell a cell if this is a self-circular RNA or not, or a foreign circular RNA? And so to test that hypothesis, we used a method to map the locations of the M6A modification on both CircFOREIGN as well as CircSELF. So there's an antibody that recognizes the M6A modification represented by the triangle. And after the antibody binds the modification, we can cross-link it, and then it digests the majority of the antibody with proteinase K. So this leaves a tiny scar of the antibody attached to the modification, which is large enough to block the reverse transcription polymerase from transcribing through that block, which means this way we can get nucleotide level resolution of exactly where the M6A modification resides. So when we did that with CircSELF and CircFOREIGN, we noticed that there was a huge M6A density peak on CircSELF that was not present in CircFOREIGN right after the splice junction. And so we also wanted to do the complementary experiment where if M6A is indeed a marker for self, that means that if we incorporate M6A into CircFOREIGN, we should be able to mask its non-selfness and decrease sort of the immune response to the transfection of those CircRNAs. And that's exactly what we found. So with unmodified CircFOREIGN, we get this high immune response. But if we just swap out 1% of the A's with M6A, we see about a 50% reduction in the immune response. And then we swap out all of the A's for M6A, that immune response is near baseline. So this suggests that M6A could indeed be this molecular marker for non-self recognition. So then we wanted to know, what are the molecular mechanisms to sense circular RNAs, and how are, these unmodified circular RNAs actually inducing this immune response? So in a previous paper from 2017, we had figured out that this innate immune sensor that's found in the cytosol, RIG-I, actually is responsible for detecting circular RNAs. Typically under a viral infection, it senses actually a 5' end of triphosphate. And so we were curious as to whether or not, this sort of unconventional sensing of circular RNAs initiated the same immune cascade as a regular sort of RNA virus with a 5' triphosphate. So how RIG-I acts is typically it's in its inactive position, but in the presence of an agonist RNA and polyubiquitin chain that stabilizes this open formation of RIG-I, which becomes activated, that can then induce filamentation or stimulation of the protein's MAVS that then have sort of downstream consequences. So we wanted to test each step along the way if that was happening once circular RNAs were detected. So we used an in vitro system to be able to specifically designate what components were going in and what was needed at which levels. So first we said, is circular RNA as an agonist RNA enough by itself to trigger RIG-I activation or filamentation? And so these are EM microscopy images where these sort of faint white lines show the RIG-I filaments that are being formed. So if you can see the filaments, that means that the agonist RNA is sufficient to sort of activate RIG-I. So in the negative control with RIG-I only, we don't see the filaments. With a positive control, we see, sort of lots of numbers of RIG-I filaments. And then in our also negative control, where we have the exact same sequence of the RNA, but it's linear and it has a 5' hydroxyl, we see essentially no filaments being formed. And then when we test our CircFOREIGN for ability to actually induce RIG-I activation, we see very few. So then we wanted to say, okay, is the polyubiquitin chain essential for RIG-I activation? And is that enough to then see sort of the next step, which is to induce MAVS to also form filaments? And again, we're using this in vitro system. So this is a native gel where you can see this band that corresponds to MAVS filaments. And as we increase the concentration of CircFOREIGN, we see a nice sort of strengthening of the number of MAVS filaments, or the concentrations of MAVS filaments. We do not see that as much with the linear RNA control. And then interestingly, when we tested our N6 modified CircRNAs at either 1% or 100%, we see a similar pattern to what we observed by qPCR. So we took these samples at the highest concentration, plated them on EM grids, and then imaged. And we see that, you know, the numbers of MAVS filaments that are being formed correlate very well to what we had observed before in the in vitro cellular system. So then we wanted to know, okay, is MAVS filamentation then sufficient to induce the next step, which is IFR3, RF3 dimerization? And indeed, CircRNA did that very potently and robustly. And we had also tested interferon. So our model at the time was that RIG-I is inactive. However, in the presence of unmodified circular RNA and the polyubiquitin chain, that can then, the RIG-I changes conformation to become active, which then sort of initiates the entire innate immune cascade. In terms of the N6 modification on the CircRNA and how that changes this process, we think that N6 can bind RIG-I, but not induce it into its active conformation. And then we have other evidence that's in the paper, but I won't have time to show today, showing that we actually, in the cells, we do need the presence of an M6 reader protein, the YTHDF2, that binds the M6 modification to, again, confer the signal of selfness that blocks this immune cascade. So then the question is, given that cells have this ability to distinguish between its own endogenous circular RNAs and foreign circular RNAs, can we use this distinction to do something useful? Can we develop a new type of therapy based on this basic science observation that we made? So we thought, could CircFOREIGN be a potent vaccine adjuvant? And we essentially took the CircFOREIGN and the model antigen of albumin, and then we injected it into mice. And then we measured the OVA-specific T-cell response and antibody titer. And we saw that the CircRNA, either with an in vivo transfection reagent or without, just by itself, was able to induce an immune response on the T-cell side that was comparable to our positive control poly-IC. We also saw that this was similar when we looked at the antibody responses. So these are all OVA-specific types of responses. So then we wondered, could we use CircFOREIGN to then prevent, say, antigen-specific tumors? Could we actually vaccinate mice against OVA-expressing cells? And so to test that, we immunized the mice, waited 2 weeks for the immune response to activate. And then we established B16 melanoma tumors that expressed ovalbumin. And then we just tracked the tumors over time. And we can see, based on the images, so the tumors are also tagged with luciferase, so we can see them on the imaging. If we compare the mice that received the CircFOREIGN vaccination compared to the negative control mice, the mice that got the vaccination were far more protected against the initiation of the tumors, as well as sort of the growth of the tumors. And in fact, this difference led to a doubling of the survival time for mice that received the CircFOREIGN compared to the mice that received just the negative control. And then because we had known about this M6A mark, we wanted to know, could we use this RNA modification as a way to tune the immunogenicity of the CircFOREIGN? And indeed, that is what we saw. So this in vivo response in terms of T cells and antibodies was also similar to what we saw with, inside cells, as well as the in vitro sort of gel and EM grid assay. So this suggests that we now have a handle of a chemical substituent that we can add into different proportions to the CircFOREIGN to then modulate the level of immune response that it's getting. So just to wrap up in the last couple of minutes, there's so many different questions that we have left to answer in this field, and it's really an exciting time to be studying circular RNAs. So in my lab, we're trying to answer as many of these questions as we can. We're interested in how does a cell know when to make a linear RNA and when to make a circular RNA. So in terms of the biogenesis, and how does the cell know, again, for these virally now identified circular RNAs, how are they different than our endogenous circular RNAs? What are the secondary structures of these circular RNAs, sort of from viral and host sources? There was a paper that came out last year suggesting that endogenous circular RNAs are more structured than their linear RNA counterparts. And we're interested in sort of getting even more molecular and understanding, what are the secondary structures, and are there unique types of secondary structures on circular RNAs? We're also interested in asking the question of, are there other chemical modifications that are present on circular RNAs, and are there other binding proteins that would recognize them? And interesting to think about would be, would there be a unique RNA modification that's present on circular RNAs that's not even found on linear RNAs? We want to know, what is the cellular trafficking of circular RNAs? We know that they're typically, well, as far as we know, they're spliced in the nucleus, and then they go into the cytoplasm, and they predominantly reside there. But the cytoplasm is a large space, and there's so many different organelles, and we know that the microenvironment of an RNA or a protein greatly affects its function. So we want to be able to establish an atlas of how circular RNAs move throughout the cell. And also, coming back to a question that was raised up earlier, how are they degraded? Circular RNAs are also packaged in vesicles and exosomes. And so could this be a way for cells that are very far apart to be able to communicate information with each other? And what is the selection for the circular RNAs, which ones get packaged and which ones do not? And then what are the circular RNAs expression and function on an organ and system level? So far, there have been many different diseases correlated with aberrant sort of expression of circular RNAs. And can we then gain a molecular understanding of exactly which circular RNAs are involved in driving, pathogenesis? And then I also want to comment on the fact that there's a strong need for technology development. For much of RNA biology's history, the focus has been on linear RNAs. And in fact, we have a lot of assumptions that all RNAs that we're interested in are linear, and they have a 5' end and 3' end. And this might be why circular RNAs were really sort of omitted and overlooked for many decades until, more recently. So so we really want to be able to develop new approaches that can target circular RNAs, as well as sort of look at linear RNAs and compare them. And then finally, can we take our basic science understanding and apply them to all of the different diseases that sort of inflict humans? And can we develop new ways of approaching therapeutic development? And could circular RNAs be a novel type of, either immune therapy or other type of treatment? So this work was really only possible due to the significant efforts of many different people. So I want to acknowledge my previous group, Howard Chang, and all the contributions there, as well as their collaborators, Bali Pulandran, Sun He, Akiko Ozaki, and Jeremy Willis. And then I started my own group at Yale last year, and it's been fantastic. And so I want to thank the people who have come through the lab and really made significant contributions. And then I also want to acknowledge our funding sources. So thank you so much for your attention. I'd be happy to take any questions.
Thank you so much, Grace. If you have some questions, I would recommend you to write it in the chat. I don't see any questions yet, but I would like to maybe start with some questions. Have you checked if the M6A modification seems to follow a pattern, like if it's more important in certain density on the circles, because I think I remember that I read that in soon, which is also a circle that seems to be modified, the M6A is in the junction as well as in the high density that you also, I think you also had in one of your analysis, like the density of M6A seems to peak in the junction. Is there like enrichment for this drag motif or so in the junction of CircRNAs?
So we have looked for first the motif throughout the whole circle, and it does not seem to be, the motif is not enriched right after the junction relative to the rest of the circle. But for some reason, you know, the modification has this increased density there. So that's definitely something that's worth digging into even more on a global level. The other thing that we did was that we had mutated individual A's so that it can no longer allow for the modification to be put on. And when we did it individually, it did not seem like there was one hotspot for modification that was required relative to the rest of this, the circle. But there's definitely, you know, like a good point that we need to do more analysis for other types of circles in addition to just like our models that we were looking at.
Okay, thank you. I think Eugenio has a question.
Yes.
Maybe you can.
So thank you, Grace, for your presentation. I just got like a long question, so that's why I'm turning my micro. So there was this group from Linglin Cheng that he described that circular RNA form a secondary structure and bind to PKR and inhibits actually their activation. So I was thinking that if this mark and will be located in those places in duplex where you find this like secondary structure, and that's why they inhibit the PKR activation.
Yeah, so that's really interesting. And they were, their story was saying that endogenous circular RNAs were binding, you know, for everybody else, bound PKR to suppress its immune sort of activation during normal states. But then under viral infection, then the circular RNAs that are bound get degraded, and then PKR is now active for sort of this rest. We really want to get into the understanding of how M6A relates to secondary structure and exactly what is that interaction. So as yet, we don't have the answer, but I think it's really interesting to consider, you know, what are the binding sites for PKR on the circular RNA? How structured are all of the circular RNAs? Are they all undergoing sort of dynamic type of movements and secondary structure changes? And where are the M6A? So super great line of questioning.
And I think I see one question in the chat from Tommaso. He asked whether RIC1 also binds linear methylated RNAs, and if this is possible, whether it's envisionable a competition between circular RNAs and endogenous as a contributor to the response.
Yeah, so RIG-I is a really interesting protein. For a long time, there was a lot of sort of interest in knowing what exactly was the motif that RIG-I recognizes. And before, people thought it was, you know, had to have a 5' triphosphate, a specific panhandle structure. It was only sort of short double-stranded RNAs, and that was it. And now we're getting a deeper and more complex understanding of how RIG-I happens. So as far as we know, RIG-I is not binding sort of endogenous linear M6A methylated RNAs. RIG-I will bind 5' triphosphate, sort of viral RNAs that are coming into the cell. And there's a group at Duke, so Stacy Horner's lab, she's been looking at, what is the M6A methylation on viral RNAs? And before, people thought that, well, M6A was a very clear mark for distinguishing between self and non-self. And she has now figured out that, well, actually viruses have sort of co-opted the system to then evade immune surveillance, but their pattern of methylation is different than sort of how endogenous circular RNAs are methylated. But again, there might be even another layer of response on the cellular side to then further distinguish between self and non-self RNAs.
Thank you very much. We have one last question, and this is from Jerry, he said, he asked if have you injected CircRNAs to any mouse model of infections, and assuming downregulation of CircRNAs, whether they might be therapeutic potential in T-cell mediated autoimmune diseases such as AAE.
Yeah, so we have so many different ideas of what we can do with the circular RNA, and we just don't have the hands yet to do the in vivo work. So we're thinking about both, models of infection as well as cancer models, different types of cancers, different types of tumors, and seeing what are the effects of circular RNAs. One might be able to imagine that circular RNAs, depending on their route of injection, may also localize to specific organs or specific parts of the body, which might be very helpful if we're thinking about therapies. And so there's a lot of room and potential for investigation here.
Definitely. Thank you very much. So